Hello students, in this video we will be talking about the DNA replication process in eukaryotes. In my previous video I have discussed about the DNA replication method in prokaryotes. Now in this video we will focus on the eukaryotic DNA replication. DNA replication in eukaryotes occur only in S phase of the cell cycle, that is also called synthesis phase of the cell cycle, that is one of the subphase of the interphase. However, pre-initiation occur in G1 phase. In eukaryotes, chromosome contains multiple origin of replication. In case of prokaryotes, there is only one site for initiating the replication process, that is ODC, the main recognition site, and after that the parts which are helping in the origination of the replication process. But in case of eukaryote, multiple origin of replication sites are present. For example, in case of yeast, ARS or autonomously replicating sequence, which is the origin site of the replication for yeast. Now, if we observe the steps of DNA replication in eukaryotes, it has three main steps like prokaryotes: initiation, extension or elongation, and termination. So first, initiation. The first step is the pre-formation of pre-initiation replication complex, which is also called pre-RC, and it occurs in two stages. First stage requires there is no CDK activities. It occurs in early G1 phase. It is known as licensing, but licensed pre-RC cannot initiate replication at G1 phase. Second stage is binding of ORC. ORC means origin recognition complex. The replication begins with binding of ORC to the origin. ORC is a hexamer of related protein and remains bounded even after DNA replication occurs. Furthermore, ORC is analog of prokaryotic DNA protein. Now, after binding of ORC to origin, CDC6, CDC18 and CDTL coordinate the loading of MCM, that means mini chromosome maintenance, to origin. And this MCM complex is thought to be major eukaryotic helicase enzyme. After binding of MCM complex to pre-RC, CDTL get displaced. Then DDK phosphorylates MCM, which activates its helicase activity. Again, DDK and CDK recruit another protein called CDC45, which then recruit all the DNA replicating protein such that the origin gets fired and replication begins. So this is about the initiation process of the DNA replication in eukaryotes. The elongation process, DNA polymerase synthesizes and adds DNTPs at 3' end of RNA primer. The leading and lagging strands are synthesized in the similar fashion as in prokaryotic DNA replication, and for this you can watch my previous video where I have discussed the leading and lagging strand formation method in prokaryotic DNA replication. So it is similar for the eukaryotic replication also here. Termination. At the end of DNA replication, the RNA primer is replaced by DNA by 5' to 3' exonuclease and polymerase activity of DNA polymerase. Exonuclease activity of DNA polymerase removes the RNA primer, and polymerase activity adds DNTPs at 3' OH ends preceding the primer. But in eukaryotic organism, okay, in case of bacteria, if we see the prokaryotic system with circular genome, the replacement of RNA primer with DNA is not a problem. because their DNA is circular DNA. But in case of eukaryotic DNA, it is a linear DNA. So in case of bacteria with circular genome, the replacement of RNA primer with DNA is not a problem because there is always a preceding 3' OH in a circular DNA. But in eukaryotic organism with linear DNA, there is a problem. When RNA primer at 5' end of dotted strand is removed, there is not a preceding 3' OH such that the DNA polymerase can use it to replace by DNA. So at 5' end of each dotted strand, there is a gap that is known as missing DNA. These missing DNA cause loss of information contained in that part, and this gap must be filled before next round of replication. So for solving this end replication problem, studies have found that linear end of DNA called telomere has GC rich repeats, and this sequence is known as telomere sequence. These repeats of telomere sequence are different among different organisms. Telomere in human cell consists of repeats of TTA, GGG and complementary base sequence AAT, CCC. Each species has its own species-specific telomere repeats. So in case of human, this is the sequence. These telomere sequence do not codes anything, but it is essential to fill the gap in dotted strand and maintain the integrity of the DNA. Telomere replication and replication problem in eukaryotic DNA. There is an enzyme found in eukaryotic cell and that is called telomerase. Here it is written telomerase enzyme. So telomere template strand, and then finally from there it replicate and form the replicated telomere. So telomerase is a DNA polymerase which adds many copies of telomere sequence at 3' OH end. You can see 3' OH end, 3' OH end of template strand. Like another DNA polymerase, telomerase also adds DNA or deoxyribonucleotide at 3' OH ends. Unlike other polymerase, telomerase adds DNA at 3' OH end of parent strand, not at the daughter strand. And also it synthesizes the same sequences over and over, absence of template strand. First, telomere binds to 3' OH ends of parent strand by hybridization between its AACCC AAC RNA sequence and TTGGGG DNA sequences. Here you can see TTGGGG DNA sequence. And this is the telomere sequence of tetrahymena. The telomerase adds TTG. Here you can see TTG is added at 3' end of parent strand. After adding TTG sequence, telomerase translocate along 5' to 3' end of parent strand. Now the telomerase adds GGG TTG to 3' end by using its CCCAAC sequence. Here in this picture you can see that again telomerase translocate and adds GGG TTA. This process is continued for many times. The parent strand become more long than daughter strand. Now RNA polymerase, which is also called primase, synthesizes RNA primer by copying the parent strand in 5' to 3' direction using telomere sequence as template. The DNA polymerase can now extend the primer in 5' to 3' direction by adding deoxyribonucleotide to 3' end. The primer is now removed and it won't be replaced because it is an extra sequence added by copying telomere sequence. And finally, the integrity of daughter strand is maintained. So this is about the DNA replication method in eukaryotes. Thank you for watching this video.